Thank you very much for coming to this seminar. It's a pleasure for me to introduce our invited speaker, that is Sahba Mobini. She's a tenured scientist at the Institute of Microelectronics here in Madrid since 2024. But before I'm going to tell you a bit about her CV. She studied in Tehran, in the Polytechnic University, and then she has done several postdocs in Iran, in UK, Germany, USA, and also in Spain. And then we are so lucky that she has joined the CSIC. She has built up a very interesting lab inside the Institute of Microelectronics based on electrical stimulation for tissue engineering and nanomedicine, and has got very important grants as Atracción de Talento, Impulsate, consolidation of the research, and she has many papers, very good citation record, and also 7 patents. Two of them granted, so it's really a pleasure that you are here, and now is your turn.
That was a rough start. Thank you very much for the kind presentation. It's an honor to be here for presenting a little bit my work on harnessing electrical cues for tissue engineering and regenerative medicine. So, as Berto said, our group called Electrical Stimulation for Tissue Engineering and Regenerative Medicine, ES4TERM, and we're based in Instituto de Micro y Nanotecnología, and we have a small group since 2020. We are part of the Campus of Excellence spiritually, but we are a little bit far in the in Tres Cantos. So I would like to a little bit start my presentation on the because I don't know how much audience are familiar with tissue engineering and regenerative medicine. So I would like to start a little bit with the general term, and then I tell you what we are really doing in this research. Basically, we all get aged, and also accidents are inevitable. Also with the escalating tension in the world, we have lots of traumatic injuries also coming from the war sources. And thanks to our lifestyle improving, and we are aging more than needed, and our species programmed to basically age, we face also problems with aging. So. Actually, regenerative medicine is addressing, aiming to repair or replace tissues and organs damaged by the disease, aging, or injury. And right now, 1 in every 3 patients needs a sort of regenerative medicine intervention. It's a big number, and also this basically requires a global market, raising the global market up to $100 billion. But what is tissue engineering? So this term is very young term. It coined in 1980s. The founders of this research area are still alive. It's basically an interdisciplinary field for replacing or substituting, restoring, and maintaining and improving tissue function. That function can be impaired by the trauma, by the injury, by cancer. But basically, tissue engineering is a way of looking to the problem, to recreating the tissue, to re-engineer the tissue. So basically, the idea behind it is saying that what is tissue consist of? Cells, a matrix that holds the cells, because cells are not there living on their own in the void, and also the signaling between cells that make them cohesively talk together and function. So what if we recreate every one of these pillars and then converge them to a living tissue? Is that possible? So it's basically a philosophy of recreating a living tissue. So for the cells, we have, since the 1980, there was lots of improvements. There were lots of ideas on using the adult cells, therapeutic cells, cells from the bank, from the tissue, from the patient, from the donors. Then come induced pluripotent stem cells, so they're getting a somatic cell, reprogram it, and make it basically to become whatever tissue we like. And also gene editing tools help these sources. As for materials, we also have lots of progress in the biomaterials, making those matrix and mimic those matrix. So using the novel chemistries that I think you guys are expert in. And then comes also using the natural materials, decellularized tissue, getting the tissue, taking the cell out, and then taking the remaining scaffold and using it as a refilling platform. And also with the 3D bioprinting and the self-assembly molecules, we also have lots of progress in this pillar. And then also comes the signaling. It's, of course, there's a biochemical talk between cells that we can imagine. There are chemical signals. There are architectural signals, so cells also sense their environment and then they respond to that environment. And there are also physical signals. And in these physical signals, we have mechanical signals, sometimes optical signals, depending on the tissue that's receiving it, and also electrical signals. So this is where we are going to get very close. But also tissue engineering also evolved since it born. So it started with creating a substitute to put back into the patient. But now people are thinking that, okay, why we can also use it as a template to make the transplanted cells more clever, or using their secretum or side material to put it back to the body. They also thought about, okay, we can also use this technique to create models so that the tissue replicates to monitor the drugs and medicine. So instead of just only thinking of making a construct to put it back into the patient body, saying that, okay, we are going to recreate human-like tissue and check the medicine on that. And also some modern applications that I don't call them science fiction applications, but they are very futuristic. So basically making artificial meat, they're all published in correct science behind it. So they're living robots, basically culturing beating cells on the flexible polymers and then making these small creatures swimming inside the culture medium. Smart tissue by combining electric platforms to the tissue, and augmentation of human capacity in terms of basically having implants, so you can also see infrared. So imagine that. So there is no barrier for imagination of these applications. So previously, I was working a lot in this material section. So we were creating these scaffolds from silk fibrines, and then we could make them nanofibrous for tissue or skin substitute. We were also creating hydrogels to facilitate combination or integration of the electrode material with the neurons. So this was part of my work in USA. And also we were working on the pattern, the structure to guide the cells to basically to change their morphology, or basically getting more closer to the niche morphology that we are targeting. But what I'm going to talk to you about, and what I have focused since 2014, is this signaling, cell signaling. And here comes the bioelectricity. So I would like to a little bit give you an introduction of what is bioelectricity. I get this gift from Smithsonian Library. I liked it a lot. This is one of the actual experiment that Giovanni Aldini, he was the nephew of Luigi Galvani. He did this experiment with the beheaded cadaver of a human prisoner. So they were like killing these prisoners with guillotine. contributing, and this guy getting this cadaver. And here in this experiment, you see that he's discharging the human body, showing that the animal electricity doesn't only exist in the frog leg, but also in humans. So it was very, very interesting. So when we are talking about bioelectricity, the first thing, the first image we normally think about is an activity of excitable cells. So very much like what we were talking about the previous slide. So these excitable cells have this capacity, thanks to voltage-gated ion channels, that they can flip the voltage of the cell membrane in a very small locality, and then that can propagate and create the neural spike, or called also action potential neural spike, which happens in the scale of milliseconds. But this is not the only, this is very well known and famous, so this created all the world of electrophysiology built around this. However, this is not the only activity, electrical activity of the cells. Non-excitable cells also have lots of electrical properties. One of the properties is this basically membrane potential that is also proven that is changing during a specific processes in the cell. So when the cell is dividing or migrating or elongating, this basically membrane potential is changing. And also every membrane organelle inside the cell have a sort of potential. This potential is maintained by the ionic current and Faradic current, thanks to the ion channels. So they're not, these cells don't have as much as voltage-gated ion channels that the neurons or the excitable cells have, but they also have their own means for basically maintaining or changing this potential during, for example, this cellular activity. One of the main cellular activity that connects to the voltage membrane is cell cycle. So during different part of cell cycle, we have different electrical properties and also cell inflammation. So again, with Luigi Galvani's sets of experiments, it's amazing. And if someone likes science history, I really recommend. It's a very good piece of work to read this summer. It is amazing what he did, and how he did is actually, he was struggling with funding. He had a funding on another project that was not get funded. He did this experiment. So it's also very interesting from point of view of the scientist and the funding struggling, that how Galvani did all this experiment. So this, his discovery of bioelectricity gave rise to the modern electrophysiology. And after that, you know, Hodgkin and Huxley make sense to basically the membrane, cell membrane conductance theory, that how this electricity creates and propagates, and how, what is basically electrical message. And then there were several Nobel Prizes in that that basically field of science, giving sense to creation, control, and sensing of electric field. This gives rise to technologies like deep brain stimulation, neural interfaces, multi-electrode array, and also nowadays biological computation. But there's this slower path, so for the non-excitable cells electricity, that, as you see, it has this also 30 years of delay in the history. So the same people who basically measured the action potential, they also measured the injury potential causing in the wound area. And that also gave some inspirations to Smith and Becker. So for the first time, they measured the exogenous electrical stimulation in mammalian tissue regeneration. And then later, people looking into how they can modulate non-electroactive cells with exogenous electrical field. And for the first time in 2010, electrical stimulation is used for manipulation of human stem cells. So as you see, it's a very, very young field of research. 2010, it was just very close. I was a PhD student in 2010. So when I was doing my postdoc in Germany, the first postdoc, I joined this lab where Ludmila Lepik was recreating Becker's experiment. It was a very interesting experiment because we were all stunned. Okay, how does this work? You cut the limb, you apply electrical stimulation, and you expect it regenerates? It's very, very questionable, because it's too good to believe. So they were basically having this idea to see that what Becker saw that encouraged him that exogenous electrical stimulation might help tissue regeneration. So they created the model of Becker, amputated the limb of the rat, applied the galvanic stimulator. It was very simple. It was 2 pieces of metal, one silver and one platinum with different connected with mega resistor, so very, very simple device. And they saw that with electrical stimulation, after 28 days, they could recruit these cartilage cells to the active cartilaginous cells to the stump. And that was very interesting because these cells are the precursors for giving rise to creation of basically bone cells. So yes, this was the recruitment of the cells, and this was rejuvenation and new cell recruitment in the stump, and also growth a little bit, but definitely not the full recreation of the limb. So we were among the first people who said that, okay, let's see with this classic idea of having scaffold cells combining with electrical stimulation, we can improve the healing, because we showed that, okay, with electrical stimulation, we can recruit proliferative cells to the basically defect area. So we generate a 5 centimeter defect, 1 centimeter defect in the femur of the rat, which is not healing on its own. So you see that this tissue, these 2 pieces of the bone that later only filled with the fibrotic tissue, but with additional of electrical stimulation, we could again recreate very much what Lepik showed in the stump of the rat. So we could recreate these cartilage cells in the site and improve basically tissue remodeling. We have done also lots of other studies in that, but we showed that 120 nanoamp of electrical stimulation, continuous electrical stimulation, would be enough to recruit cells in the defect area. But the things are still, as I said, compared to the faster cousin, the electrophysiology is much slower. We don't have too much of FDA-approved products on that, and most of the work has been done is around skin and bone, and the most challengeable tissues like spinal cord, neural regeneration, cardiac healing is still under question. And we want to know why. Where is the bottleneck in this research area? So we think that right now we don't have a standard protocols for electrical stimulation. There is no way of consistent reporting of how much electricity that we are really delivering to the cells, because whatever people are reporting is that, oh, I attached this to this battery or 2 volts, but really how much cells are sensing in the place is still not reporting very well, and there are also lack of translatable methods to apply electrical stimulation for doing research. the nature of the response, the readout is slow. So what we are reading is very slow, and it's very difficult to differentiate it from the other processes in the cell. So it's not like action potential. There is a spike, and you measure it and say that, yes, I got it. You need to wait 3 days and see that, okay, now what I'm reading. So it's very difficult. The nature of the response is slow. We are still not sure if the nature of response is all or none. So are we really creating a one-to-one response to electrical stimulation when we are dealing with the slow stimulation, or it is something like more universal? So this is still under not clear. So basically, this leads to unclear dominating biophysical mechanisms, and also, in the end, if you want to use it, it's invasive. Oh, okay, so a little bit translation point of view. So in my lab, for now, we are focusing on some of these problems. Problem one, we need platform. We need protocol to apply electrical stimulation. So there are several ways you can imagine you can apply electrical stimulation. It can be inductive, that you basically are using capacitive plates or inductive electromagnetic field, or you can just deep two electrodes inside the culture and create electric field. We are interested in the direct electrical stimulation. Why? Oh, sorry. I think something lost in translation with the... Sorry about the quality of the image. It was not like that. So anyway, but I think you can imagine. So because we want to recreate the sort of in vivo electrical stimulation with the probes, and here we know that when we are using direct electrical stimulation, we are dealing not only with an electric basically environment, we are dealing with electrochemical environment. So it's very important to understand this, to model that. So we are using random models to basically translate what is happening in the interface of electrode-electrolyte to give some sense that finally what cells are sensing. I apologize for the quality. So in our lab, we are also interested in creating these electrodes. We are also interested in creating electrodes with augmented electrochemical efficiency. That is why we are searching for nanostructuration solutions. This is basically where the tie between me and our institute, that we are developing these nanoelectrodes, electrodes with nano roughness, and basically we show that we can increase this effective surface area that later translates to better, higher charge injection capacity and lower impedance. And we tested several metals for that, and finally we stick with platinum electrodes that we show that also with the nanostructuration we can decrease their impedance, increase their charge injection capacity, reduce the Faraday byproduct they are inserting to the cell culture. We can keep the delivery basically signal very close to applied signal, so we have less reduction in the attenuation in the delivered signal, and we also develop these devices, testing them in the in vitro settings and simulated in vivo settings, and we show that they are quite stable. So we created these devices with fully characterized, electrochemically characterized, and we didn't stop here. These devices are basically expanded to the several platforms that you could see here, the platforms that biologists like to use, 24 well plates, 84 well plates. So we have all these type of basically stimulators. It's very difficult to present like this, but I try. We also created a monitoring system to do in situ corona amperometry, so we can know that how much cells are receiving current in the time of stimulation. And also using equivalent circuit, we could simulate exactly what is happening in the virtual model. So we basically have our digital twin for every stimulation device that we have. You can see one of our devices in the work. So this is the stimulation module, basically, and this is our cell culture module. So we have the cells here, and we apply electrical stimulation with this device, and we could do them in basically in several stimulation in the same time. And we can do the stimulation inside the incubator. We can monitor exactly what is happening in the cell, in the cell culture area. And we could basically apply several signals, and we can control them very effectively. Okay, but what we are using this technology for, one of the applications that we are interested in is neural maturation. Neural maturation is an important topic in tissue engineering and regenerative medicine for fundamental biological mechanistic study. If we know that if we create a model that is a mature neuron, it's a very important, basically, asset. Why? Because normally, the neurons, especially neurons from human that we are working with them in the lab, they're very young. They're either coming from embryonic stem cells or they are coming from differentiated from the iPSC cells. So these are resembling the neonatal neurons. And so imagine we want to create an Alzheimer model. An Alzheimer model will never be able to create it in the young neuron. So you need to age the neuron, but we cannot culture that neuron 70 years. So how is that possible? We need an accelerator for neural maturation. And this is one of the questions in the field of modeling neural and tissue disease, that we are also interested in accelerating this maturation with electrical stimulation, so exercising these neurons. And also, it's important if we can find a solution for disease. We know that electrical stimulation, so normally for the neural differentiation and maturation, people are using retinoic acid, BDNF, that is basically kicks the differentiation mechanism and then later maturation. And then we think that with electrical stimulation, we can tap some of these mechanisms. But the question is that with which electrical stimulation paradigm are we able to kick on the maturation? So that's a very, very good question. So many people are starting to play with the voltages, amplitude, and pulse duration, but you can imagine that you have infinite number of combination for these regimes that you can apply to your cell culture. But we changed a little bit of how we are looking at this question. Instead of making ourselves crazy with the infinite table of combination of frequency, amplitude, pulse width, we focused on the physical implication of this electrical stimulation. So how much charge we are putting in the system and how much electric field. And also we used this cyclic voltammetry measurement to find out where basically is the safe voltage window, and we are not splitting water. So we set ourselves there. So in one of our important experiments, we could design different electrical stimulation with different charge injection capacity. So we have a stimulation 1 with a 0.1 and a stimulation 3 with 50 basically millicolon in 1 hour injection, and we wanted to basically measure the effect of discharge injection capacity. We also designed a sort of brave experiment in the sense that we wanted to see that if electrical stimulation can replicate the effect of endogenous BDNF, basically the chemical people use for creating neural maturation. So these are our control groups, no electrical stimulation and no BDNF, and this is no electrical stimulation BDNF, so this is our positive group. And then with electrical stimulation, with increase of the charge injection, we show that even with the absence of BDNF, we could recreate the effect of maturation. So that was very, very impressive, and we show that this is somehow connected to the amount of charge we are injecting to the system. We also showed that it is not only about the morphology of these cells. We can increase the synaptic markers and also neural markers, tau5. And also, we looked into cell proliferation and DNA synthesis that was basically corroborating our results for the maturation, because it's basically a seesaw response. So we basically showed that the biological outputs of the neural differentiation and maturation are likely controllable with exogenous electrical stimulation, and probably it can be controlled by the input charge. And electrical stimulation can replace exogenous BDNF use. That's a very good news for biologists that want to look into the effect of some pharmaceuticals that they don't want to have the additional BDNF in their system. So basically, you can create something with a chemical-free or semi-chemical-free environment. We didn't stop here, so we said that, okay, so now we need to see that what is how we can use this technology. So as you may be aware of, the human cerebral organoids are now becoming very important in terms of modeling in vitro. So they are these three-dimensional, basically multicellular structures that are able to mimic, hopefully, the human physiology for modeling the disease. However, they're also challenged by neural maturation. So they are very young. So then the neurons that are very young, and also they can suffer from the necrotic core, because you cannot basically grow them bigger enough. So we also applied electrical stimulation to these human cerebral organoids, come from the human iPSC cells. This is done by our collaborator in our collaboration with Ira Espuni Camacho in Giga Stem Cell Institute in Liège University. So they have these super interesting cerebral organoids, and we use them to see if electrical stimulation can improve their maturation. We showed that we could successfully, I really apologize for the quality of slides, it's not my fault. We show that with electrical stimulation we can improve some of the neuronal markers, and also rosette counts. Rosettes are these cerebral structures that some of the like organized structures in the cerebral organoids that are very important as a characteristics of their maturation. And so we basically showed that electrical stimulation can be a promising tool for enhancing this maturation. And now our national project grant for the next 3 years dedicated to hash out all the details about these promising initial results. We also collaborate with Sydney University, with Anaya González Cordero. They have these systems for retina regeneration with electrical stimulation, and we are basically We are basically providing them with a stimulation platform that they can put their retina organoids there and electrically stimulate them. And they also showed very similar parallel results to our cerebral organoids. We are also using, so when we see that electrical stimulation can benefit the BDNF creation pathway, we also looked into the recovery, basically effect of electrical stimulation. We create currently this stroke model, excitotoxic model, and we are looking into different, basically, application of electrical stimulation in reversal of apoptosis. This is the work that is currently carrying out by Miriam Istasi, our postdoc, and also Gaurav Kulkarni. We create these injured neurons, so we get the neurons, we differentiate them with BDNF, and then we apply neural injury with excess of glutamate. This injury, we basically characterize the injury with the live and dead cell assay, with looking into different degree of the apoptosis population, and also chromatin compaction and expression of caspase and BAX-BCL as a marker of different stages of apoptosis. So currently we have this model in our lab, and we are applying electrical stimulation. So our first kickoff experiment, we could show that we could reverse basically apoptosis by showing a reduction in the number of the apoptotic positive cells, and also by showing the number of survived cells. So LDH is the component that releases from the dead cells, and as you see here, in the non-injury, no electrical stimulation, completely a control group, we have very low release. When we have the injury, this release basically, when we have no injury and electrical stimulation, we also don't kill the cells. When we have the injury and no stimulation, this component releases more. And when we have the injury treated with electrical stimulation, we reverse them back to more viable cells. So this is one of our ongoing projects. Another very interesting line that we are working on is the cell secretome. This is the final part of my talk. I will finish in 5 minutes. I don't know if how much you are familiar with the cell secretome. So cell secretome is basically the secreted material from the cells that previously thought they are basically bag of garbage that cell will put every night outside. But then later they find out that no, they have lots of implications. So they lots of cell-cell crosstalk and also messaging, basically exchanging between the cells within this secretome. What is very interesting is that secretome has 2 fractions, a soluble fraction, so proteins that are soluble in the soup, and also a fraction that is non-soluble, and that these are these nanoparticles, natural nanoparticles that is consist of lipid bilayer, and they contain biomolecules, proteins, mRNA, siRNA, and these are very important for cell-cell signaling. They can be created from different, in different ways. So we have these exosomes that are creating through the inward budding of the membrane, and then later they create these multivesicular bodies. They basically exchange their cargo, so what they bring from outside, they release it, and they also repackage new cargo from the protein synthesizes in the cell, and then they release those exosomes. And then these exosomes will be uptaken by the recipient cells and changing these messages between cells. These exosomes, natural nanoparticles. They basically express several proteins that are in common with the recipient and the producer cell, and that is why it makes it much easier for the uptake through the recipient cells. And also they carry protein, RNA, amino acid, and metabolites. It's a huge world now created around the extracellular vesicles. There is a path that many people are focusing on using these extracellular vesicles as a diagnostic tool, so using them to read what is happening. It's normally also cancer cells also producing these extracellular vesicles, and when they are looking into those vesicles as an early reporter of the presence of cancer in the body, but also many people looking into the therapeutic effect of these vesicles. These vesicles are very interesting in the sense that they can be uptaken because of their friendly membrane, and they can also pass natural barriers like blood-brain barriers. So they're very, very good candidate for the therapy. And that's why now many people are thinking that perhaps cell therapy one day will replace by extracellular vesicle therapy. And why? Because they find out also in a very parallel line of research that in the cell transplantation, we end up not having the cells differentiated to the line that we are expecting to. So cells also filter through the lung and liver and our kidneys. So after 48 hours, cell transplantation, nothing is in the body. But still, there are some effects. And that is the smoking gun, that thinking that this effect is coming from, this effect coming from basically paracrine signaling, not the cell itself. So this made the community become very, very interested in the cell therapy rather than in the exosome therapy rather than cell therapy, and also looking into how they can make it better. Because there are two problems with these EVs. It's very difficult to produce and purify them. The yield of production is very low. And also, we don't have control on the cargo packaging when they are in the native state. So there's lots of effort coming from the nanoparticle world that they would like to functionalize it, to use the vehicles, to basically post-engineering these EVs. And there's also one idea of, instead of post-engineering, conditioning the mother cell and giving rise to the EVs with better basically qualifications, and also increasing the yield of application. So the idea of creating these these EVs with electrical stimulation come from one of my patents in United States in 2018. We did this experiment. It was very interesting. We got this human adipose-derived mesenchymal stem cells, applied electrical stimulation to them, and we take the secretome and just load it on the gel, and you can just see that we improved amount of proteins. So this was the first thing. There are lots of proteins there. Then we did a deep proteomics study on them, and we find out that not only we increase the amount of proteins, we change the signature of these proteins, and it also depends on the electrical stimulation that we are applying. The effect of this secretome later, when we applied on Huvik cells, is a model for creating vasculature in the in vitro, was also different, and it depends on the how we treated these mother cells. So that was a very interesting finding. So when I come to Spain and this world around the extracellular vesicles were created, and they were saying that the effect of secretome is basically connected to the extracellular vesicles. We focused on that, so we repeated the experiment. We get the electrically stimulated secretome. We purified the EVs, and this time we showed that we also can increase the amount of EVs and also the protein content, both of them. And we applied it on our human cerebral organoid model. We showed that we can accelerate the growth rate of these organoids. So this was a very interesting pilot study, but we couldn't repeat it with this exosomes coming from another cell source. So these exosomes coming from the bone marrow-derived mesenchymal stem cells, but when we changed the cell source, just the same cells coming from another tissue, we couldn't repeat the effect. So that started to make everything much more interesting, saying that this, and also we couldn't repeat the effect when we were working with the older passages. So we see that everything is very, very sensitive. So we went back and we tried to use some standard cells. Now we are working with these Eversite cells that are coming with a telomerase modification, that they don't become senescent. So we can use every passage and we can create much more uniform exosomes. And we could have them from both tissues. So the study I showed you previously, they're coming from human donors that we get, one of them from Complutense University and the other one from Zaragoza University. But these ones are completely identified cells. So we could improve the secretion of the secretome with electrical stimulation and augmented the number of the particles that we could identify as EVs, while we were basically inducing no changes in the size. We could also see that the signature of the proteome for these EVs are different under electrical stimulation, though they are expressing 90% of the exosomal signature, so they are very also natural, so we are not changing them a lot. So we are now patenting this method. We are under the path of patenting this method and developing a spin-off company for creating these exosomes. We also did another deep proteomics study, and we find out that in the BMMSCs, which we're working for our organoids, what we can increase is the family of the proteins that are connected to neural development and plasticity. So it's also a little bit answered our doubt before why the EVs coming from the adipose tissue didn't have the same effect, and we kind of found out that we increasing the neuroprogenitor protein families in those EVs. So with that, I would like to summarize my talk, which also took a little bit long. So we created we developed these highly versatile, reproducible platforms for electrical stimulation. We have 2D and 3D models showing that we can use this electrical stimulation for neural maturation and reversal of the injury. These are under study, and also we can use them for creating extracellular vesicles. So with that, I would like to thank my team and my funding and my collaborators. Thank you. And sorry about the quality of the presentation. It was not much.
Don't worry, the presentation was great. Thank you very much, Sahba. It was a very nice work. I hope there are many questions. Anna?
Thank you very much. Sorry. Yeah, very promising. I have a question regarding, have you ever detected an increase of temperature while you apply electrical stimulation?
Okay, so if you remember, I showed a cyclic voltammetry curve that we were doing. So because it's we never measured that directly, because also the temperature changes. We believe that there is a nanojoule changes in the temperature, definitely we think that it is happening. It's also some of our models showing that, but we never measure that. One important reason is that we didn't have the tool.
Yes.
A reporter, a nano temperature reporter, and I see that there are lots of beautiful works in this institute that might be super relevant. So we didn't have that reporter, and also, but we measured, for example, the pH. The pH, if we are inside the safe, let's say, voltage window or limit, we can. The pH change is basically irrelevant because everything is also happening in a very strong buffer. So when we are working with the cell culture medium, this is a very strong buffer, and that can basically cover all these small changes in the pH. Yeah, that is my answer to your
Very
question.
interesting. Thank you very much.
Thank you very much for the talk. It's amazing work. So I would like to ask you about the last part of the talk, that you said it was related with plasticity, the results that you found. And this is also curiosity. So in these organoids, have they, someone have measured neurotransmitters that are related with plasticity, like dopamine, serotonin, GABA?
Yeah. So and our partner in the Giga neuroscience, Giga stem cell, it's a huge facility that there are only cell culture rooms with separate incubators. They are looking into developing really old organoids for resembling the Alzheimer. So they are measuring these things in these neurotransmitters on the regular basis, because this is how they are creating this model. So we started with the electrical stimulation of these organoids in a year and a half ago. We just wanted to see that if at all it is relevant, and when we see that it is relevant now, our postdoc is going to stay there for 3 months, starting from September, and now we want to do more pilots. So we do more basically replicates, and also go deeper to see that what we are really changing that results in maturation. Yes, this is, of course, this is a very, very interesting way to look at.
Thank you very much, Sahba, for this impressive work. A lot of ideas to assimilate. I have a question concerning when you show us the fluorescent images. So these are from DAPI or cells that are staying with DAPI or whatever. So these are done in situ, or you just electrically stimulate the samples and then later on you observe them under the fluorescent microscope?
Okay, yes, sorry, it was not enough time. I go to my graphs like a good student. I present very bad, so I'm going to walk you through these images. These are images from the fixed organoids. So if you see here, oh, no, I don't have it, I don't have the, so when we are doing the, after electrical stimulation or after application of the EVs, we fix the organoids in the OCT.
So it's clear, it's not in situ.
No,
so you don't observe this in situ.
Yes, this is not in situ. But, and the markers here you can see are the markers that are reporting the cell cycle or amplification of DNA from the, so the Chi, for example, is a reporter for the amplification of the DNA. So these are the reporters that we can read them after everything and on the fixed. For the in situ, this is also one of the experiments we designed to be done in September, but we would like to look at the calcium, basically lighting up directly with electrical stimulation, but it is very, very, very tricky because these are three-dimensional, and we need a very good confocal microscope. So technically it's a very difficult thing to do.
Okay. More questions? I have one. How local is the stimulation that you apply? Because you do it with 2 electrodes. I see the system that you develop. how far is going from the electrode, the stimulation?
The short answer is that we are applying bulk electric field. So it's basically what we are stimulating cells with is the electric field and also the gradient of the charge. So it is not local, so it's not cell by cell control. And this reactor that you saw, the distance between the two electrodes is 1 centimeter.
Okay. And it's important the direction?
Yes, it is important. The direction depends on what you're looking at. If you are looking at migration, direction is absolutely important. But now we are, for example, working on the biphasic stimulation, so we cancel the preference of the direction. So with that, we can only look into a specific way. It is very difficult to separate the components of electrical stimulation. That is one of our challenges. But we are trying to find out ways to design experiments that we try to look at one specific things, and this is one of the tricks.
Very good. Thank you. Very interesting. Any other question? No? So let me thank the speaker again. Thank you very much, Sahba.